And we're here. And we were looking at mechanisms for the evolution of different types of mate choice. Why do mates choose their mates based on particular traits? What is it about those traits that makes them the target of sexual selection? And so we looked at the good genes hypothesis, we looked at hybridization avoidance, we looked at possible reasons for assortative and disortative mating. In some cases, it just seems to be pre-existing sensory bias, that there's something about the way that an animal's nervous system is wired up that causes it to prefer mates of one type or another. And this was worked out by the work about 25 years ago of a biologist at University of Nebraska named Alexandra Basolo, working on a group of fish called swordtails in the genus Xiphophorus. Xiphophorus, by the way, not that you need to know, but that happens to be Greek for sword bearer. This is a pair of Xiphophorus multilineatus. The female, you can hardly see it, but she's got pretty much an ordinary fish-type tail. The male has a more or less ordinary fish-type tail, except down here on the lower margin, it has this long extension. This extension looks like the male is carrying a sword, although in fact it's not sharp. It's flexible, it's just elongated fin rays. The male can't stab with it or anything like that. And there's about 20 odd species in the genus Xiphophorus. They're native mostly to Mexico and Central America. Some of them are popular in the home aquarium trade. This is the green swordtail Xiphophorus helleri, and that's a male, of course, and most of his tail is translucent. It's right there. But you can see this very prominent gold sword with a black edge extending out like that. Females, by the way, never have swords, and females like males with big swords. What makes Xiphophorus an interesting kind of natural laboratory to study sexual selection is that some species of Xiphophorus have males with long swords. Some species, the male's got short swords. In other species, the male has no sword at all. This is a male, Xiphophorus maculatus, and he's a male. You can tell, by the way, because he's got a rolled up pelvic fin right here. That's called his gonopodium. That's a sperm transfer organ. So you can tell he's a male, but his tail just comes out like that, and he doesn't have a sword at all. So X helleri, males have long swords. X maculatus, males don't have swords. Now, Basolo did some brilliant experimental work on preferences that involved doing things like she could cut artificial swords out of plastic and give males longer swords by sewing these plastic swords onto the male's tail. She could give males from swordless species artificial swords that they'd never had before. She was able to do all kinds of experiments by adding swords and manipulating sword length. And she showed first that, as you might expect, females of species with swords preferred males with swords. Female Xiphophorus helleri with the long sword, the females preferred males with long swords, not males with short swords or no swords. But the preference existed in the swordless species as well. Female platyfish, when given a choice between a normal male of their species and a male with an artificial sword stitched on, there's an artificial sword that's been sewn on. There's a male with, implants, I guess. Females preferred males with swords, even though they would never encounter these in their natural habitat because the males were swordless. And she found that that preference of females evolved much earlier than the trait ever did. The females had the preference before the males evolved the trait. And what seems to be going on is that female fish aren't really rocket scientists. They're not really brilliant. And what they really prefer is males that are large. They'll go for big males over small males, possibly because maybe the big males have better genes. Um, I don't really, maybe the big males produce more sperm or have higher quality offspring. I don't really know. But females prefer big males, and they're not really good at assessing the size of a male. They just kind of compute overall length. And in some species of Xiphophorus, the males evolved swords because males that had swords looked like they were bigger, even though their bodies weren't necessarily bigger. If they looked bigger, they got more mates than if they didn't. So in this case, the preference is older than the trait. The trait evolved in some species because the preference was already there. The trait evolved because having a sword makes a male fish look larger. The final model we'll talk about is called runaway sexual selection, and runaway selection is what you get when you have selection on a trait for no other reason than the trait is there. In runaway selection, there's no inherent reason why a particular trait becomes the target of sexual selection. And that sounds weird, but a great example of it is Diabrotica undecimpunctata, the spotted cucumber beetle. What they found when looking at spotted cucumber beetles is that before mating, males stroke the females with their antennae. The males will just hang out right next to the females and reach those antennae over and give them a good stroking. That sounded weird. Why would they be doing this? Well, it turns out that none of the other explanations work. Males that stroke females don't have higher quality offspring than males that don't. There's no obvious good genes advantage. There's no, the offspring of long stroking males don't survive any better or worse than short stroking males. It's not a marker of genetic quality. It's not a handicap. There doesn't seem to be any logical reason for the preference, except let's say we have males that have some kind of trait that varies, and we have females that vary in their degree of preference for this trait. So let's say we have long strokers and short strokers among the males. Could you close the door, please? Please keep the cool air inside. All right. So we have males that are short strokers and males that are long strokers. And let's say we have females that vary in their degree of preference. Some females like long strokers, some like short strokers. So the females with the strong preference choose males with the strong trait. Females that prefer lots of stroking will mate with males that stroke long. And over time, the genes for the trait and the genes for the preference will behave as if they're linked. They're not necessarily physically linked on a chromosome, but they will be inherited together because every beetle that inherits the trait of long stroking, comes from a female parent that has the preference for long stroking. So the trait and the preference tend to go together.
I have an example.
They will act as if they are genetically linked, even though they may not necessarily be physically linked. They will occur together much more frequently than you would expect just by independent assortment. So then anything that happens to favor one to become more common will cause the other to become more common. It could be a little thing. Let's say there is some predator that comes along and it just happens to kill off a disproportionately large number of the short stroking males. Well, then the females that prefer the short strokers can't find anybody to mate with. Females that prefer the long strokers have more mates and will leave more offspring. Okay, one moment, folks. I need to pause the recording. Boom. Okay, so let's say that something happens to kill off more of the short stroked males than the long stroking males. Purely by chance, it could be. If that's the case, then there's fewer short stroking males. So the females that prefer short stroking males don't have as many mates and they don't leave as many offspring. Females that prefer long stroking males have more mates and they'll leave more offspring. And those offspring, remember, will inherit both the long stroking and the preference for long stroking. So that means that females that have the preference for long stroking, they'll find more mates. Males that stroke long will find more mates. As the trait becomes more common, the preference becomes more common. And as the preference becomes more common, the trait becomes more common. Each one tends to drive the other. So it's kind of a positive feedback situation. The trait increases in frequency because the preference increased in frequency because the trait increased in frequency because the preference increased in frequency. That's runaway sexual selection. Under the right conditions, this can happen, where a trait that confers no benefit on either the male or the female, it's not a marker of good genes, it's not a sign of genetic quality, it's not a handicap, it's not anything we've talked about, except that females that have the preference for the sexy trait will tend to have sons that that show the sexy trait. This is sometimes called the sexy sons hypothesis. And this was shown in 2001 in the study of female spotted cucumber beetles. The females don't benefit at all from being stroked by the male's antennae. The offspring are at no advantage. The offspring of fast stroking males are not more or less likely to survive or reproduce than the offspring of slow stroking males. The only difference is that fast stroking males tend to have fast stroking sons, and females prefer to mate with those. It's a trait that becomes more popular because it's more popular. Okay, that may be a little counterintuitive, but that's runaway sexual selection. And this is one aspect of the sexy sons hypothesis, which one of the architects of the modern synthesis, a guy named Ronald Fisher, proposed in 1930. A female who picks a male that has a desirable trait will leave male offspring that have the desirable trait. And it doesn't even matter what the trait is. The trait could be something like eyes on ridiculously long eye stalks, like this male Malaysian stalk-eyed fly. That's a fly. And instead of the eyes being on the head the way they would be in houseflies, the eyes are way the heck out on stalks. And this seems to be a similar case where there are females that will prefer either short or long stalked flies and males that have either short or long stalks. And the genes for the trait and the genes for the preference act as if they are linked, which means that anything that increases the frequency of one automatically increases the frequency of the other. So you get this, that's runaway sexual selection. Whew, oofda. Okay, now I've introduced the idea of selection acting not only on physical traits, but on behavior patterns. We're going to transition to a slightly different subject that I'm going to jam on, and that is evolutionary altruism. Consider, if you will, um, any of you that have worked with Dr. Hazelcorn will have heard about these. These are dictyostelid slime molds. Most of the life cycle is spent as single-celled organisms, little amoebas. If you remember, an amoeba is a eukaryotic cell that doesn't have a fixed shape and gets around by flowing from place to place. So here's some little dictyostelid amoebas crawling around in soil, in leaf litter, feeding on bacteria and just doing their little amoeba thing until food begins to grow scarce. Under starvation conditions, these amoebae start making like John Lennon and coming together. Get it? He was a member of this band called the Beatles that we used to have back in the old days, and sang this song called Come Together Right Now Over Me. Well, here are a bunch of amoebas that are coming together right now over me, aggregating into these clusters. So that's a big cluster of amoebas that have all assembled. And these clusters will get large enough, they'll form millimeter scale slugs that may crawl around for some time. They look like a multi-celled animal. I guess you could call it that, although it's kind of an odd way to think of multicellularity. But the slug will crawl around for a while, and eventually the slug will grow into a stalked sporangium. You actually have immature stages over here on the left, and more mature ones over to the right. And then over here at the far right, this thing with a very slender stalk and a big tether ball on it, that is a mature sporangium. Okay. What happens is that cells in this upper sphere, they grow cell walls around themselves and they form spores. The sporangium up here will dry out and the spores will go drifting away on the wind. And eventually, if all goes well, they'll land in a new habitat. The spores will open, the amoebas will come out, and the amoebas will resume their life cycle, crawling around in soils and leaf litters, um, feeding on bacteria. Here's where it gets weird. That's a diagram from the work of one of the earliest people to study these from an evolutionary point of view, a great biologist and essayist named John Tyler Bonner. And this is an early stage in the development of the sporangium. The sporangium has a stalk, and the stalk is attached to an attachment disc. And the weird thing is that the stalk and the attachment disc are made of amoebas. But what the amoebas do is lock together, grow these cellulose cell walls, and then they die. So if you are an amoeba in one of these slugs, okay, one of these aggregates right there, if you end up in the sporangium, if you end up here, if you end up here, you will form a spore and float away, and you've got a chance of surviving. But if you end up in the stalk, there, or in the little holdfast down here, you have no chance of surviving. You're dead. You will give up your life. If you didn't do that, then these spores would not be able to get up into the air column and they wouldn't be able to disperse. So if you're one of these amoebas in the stalk, you are enabling these spores to survive, but you yourself are dying. You yourself are dead. And that's really weird. We can think of why humans might sometimes choose on the basis of their values and their culture to sacrifice their own lives for their neighbors. But these are amoebas we're talking about. They don't think, they haven't got brains. They're not capable of saying, I am prepared to give my life so that the greater slime mold colony may survive. I regret that I have only one life to give for my species. No, amoebas can't say that because amoebas don't have brains. Amoebas just do what amoebas do. This is an extreme example of altruism, the giving up of a benefit um, for the sake of other members of your species. And it raises a bit of a conundrum for natural selection because why would selection favor individuals that don't pass their genes on at all? If you're an amoeba and you've got genes that in some way cause you to end up as part of the stalk, well, those genes have just hit a dead end because you're going to die as part of the stalk and you will never pass those genes on. So, how could genes for being in the stalk ever evolve in the first place? We do know, by the way, that there are genes with this effect because it's been possible in the laboratory to create stalkless mutants, mutants of cellular slime molds that don't build stalks because every amoeba gets to form a spore. So we know there's a genetic basis for this, but how could that genetic basis ever evolve? Last I looked, we don't really have a full answer for slime molds, and maybe on Friday I'll try to come back and see if I can address what we know about slime molds. But let's look at the more general puzzle. Altruism is what you get when two organisms interact in such a way that one receives a benefit and the other seemingly gives up a benefit. Very often, that benefit takes the form of a gain in fitness. Now, you'll find in old books that individuals can evolve to sacrifice themselves for the good of the species, but under the standard model of natural selection, this doesn't seem possible. How could genes ever become more common if the effect of those genes is to block themselves from getting passed on? Well, a fellow by the name of Richard Dawkins wrote a book in 1976, still worth reading, called The Selfish Gene. Um, Dawkins put a lot of this together, but many of the ideas were not original to him. They'd been developed by biologists like George Williams and especially William Hamilton. But it was Dawkins that put a lot of the pieces together in an engaging way and came up with a metaphor for what we're talking about, the metaphor of the selfish gene. Here's the thing, there's really no such thing as a selfish gene because a gene cannot be selfish or unselfish because a gene is just a string of nucleotides. Right? Your genes can't actually want anything or desire anything because a gene is basically a very complicated chemical compound. So a gene just does what it does, and genes don't really have motivations. So genes aren't really selfish, but talking about them as if they were can help you grasp the issues. Williams and Hamilton had argued that selection could favor altruism if it was for the good of the group, that organisms could commit altruism if it caused the group to have a better chance of survival. Williams and Hamilton argued that's not possible. Let me show you before we move on why it's not possible. Imagine you've got a town full of altruists, full of people who will always help each other out no matter what. They will lend you the shirt off your back if you need it. They'll lend you all the money that you want if you need it. They're always helping each other, always, never holding back. They are perfectly altruistic towards each other. one criminal is going to clean out that town completely because a society of pure altruists has no way of screening against people that take advantage of them. And that's the idea. If you had a species where every member of the population was perfectly altruistic, one mutant that wasn't altruistic ought to reproduce because it takes everybody's resources, and that ought to reproduce and spread very quickly. That ought to be favored. So altruism simply for the good of the species or the good of the population, the good of the group, doesn't actually work. The selfish gene model looks like this. Genes are the fundamental units of selection. That what selection fundamentally does is make genes become more common or more rare. The genes that are favored by selection are the ones that have an effect that maximize their own probability of replication. Right? If a gene has an effect on phenotype, that effect might increase the gene's own probability of getting passed on. And if a gene has that effect, then it will become more common in the population. Those effects on phenotype don't always appear favorable to the organism that carries the genes. They may look as if they are causing the organism that carries those genes to have reduced fitness, but that can be deceiving. Genes can have effects on organisms that make it look like they're giving up fitness while actually increasing the probability that the genes will get passed on to the next generation. Just selection can look one way when you look at organisms and their phenotypes, and a different way when you look at the effect on the frequency of genes. So genes aren't really selfish, but genes look as if they're acting to maximize their own chances of replication, even if this does not look like it's in the interests of the organism that's got them. That's what we mean by selfish genes. So let's look at some examples of this. How do you get the evolution of altruistic behavior? Why should organisms evolve in such a way that they seemingly give up benefits to each other? The simplest example are cases where individuals have no chance to survive unless they're altruistic. There are cases where altruistic behaviors may be the only solution, where not being altruistic just means you're dead, where organisms will cooperate with each other and give benefits to each other because there is no other way. This is the making the best of it hypothesis. You get this in organisms where the environment is harsh and where they have a dominance hierarchy. And the Galapagos hawk turns out to be a great example. You may remember from way back in towards the beginning that this is a species of hawk. It's related to North American buteos. It's related to the red-tailed hawk and red-shouldered hawk that you can see around here most years. But it's not quite the same. It's one of these species that is like mainland species and yet unique to the Galapagos. But there's more to it than that. The Galapagos hawk shows this rather odd mating system. It's polyandrous. What you have is stable groups of unrelated males that all mate with a single female, and they all share parental care. This seems altruistic because every male hawk has fewer chances to pass on his genes than he would if he could pair up exclusively with the female. If a male hawk could find a female that would be all his own, he could fertilize all of her eggs and be the father to all of her babies. Whereas if he has to share a female with, I don't know, 3 other male hawks, then only a quarter of her offspring are going to be his. So by putting up with this, he seems like he is giving up a fitness benefit. He can't have as many offspring by sharing a female than he would if he had a female all his own. So that's altruism, where the males are seemingly giving up a benefit. The reason why it makes sense is, remember that the Galapagos are not these tropical paradises um, where waiters bring you fruity rum drinks with paper umbrellas. The Galapagos Islands are desert. Some years there's more rain than others. There can be, absolutely vicious drought, especially in El Nino years. And that means there's not a huge amount of food. Since food is sparse, Buteo has to defend large territories to survive. They've got to stake out claims for a territory where they can hunt and defend those claims against other groups. And it turns out that under those rather harsh circumstances, males cooperate because there's no other way. A group of males can defend a high quality territory with good hunting. A lone pair of hawks can't do that. A lone pair of hawks trying to defend a territory is going to get sand kicked in its face, is going to get its lunch money stolen, and is going to wind up stuffed in the gym locker. Okay, not literally, but figuratively speaking, that's what's going to happen to a lone pair. A lone pair can't survive at all because a lone pair can always be kicked out of its territory by a group. So a male hawk that preferred having a female all its own is not going to leave many offspring at all because it's not going to be able to find any food. A male hawk that gives up a benefit in the form of not having as many offspring as it could, those males are going to survive and pass their genes on. So sometimes altruism is the only way for a population to survive in the first place. Dawkins came up with another example. This is the last thing I'm going to talk about before break, called the green beard effect. He's very good at coming up with catchy names. So imagine you've got a gene, or maybe a couple of tightly linked genes, doesn't really matter, that confer a trait on individuals that have it and cause individuals that have that trait to behave altruistically towards other individuals that have it. Dawkins said, imagine you have a gene that, first of all, if you have it, you grow a green beard. And the other effect of that gene is you not only grow a green beard, you are drawn to behave altruistically towards other individuals who have green beards. So if you have a green beard, you help other individuals with green beards, and those other individuals help you. So imagine you've got a gene that both confers a trait and encourages altruism among individuals that have that very trait. If that appears, then both the trait and the altruism should become more common in the population. That's weird, and it's hard to think about when you're only thinking of it as a theoretical possibility, but this was a real world example from about 15 years ago, and it's found in brewer's yeast, Saccharomyces cerevisiae. Very, very, very common organism for cell biology research. And the strains that have been traditionally used in cell biology research, the little cells are suspended and they don't clump together. The strains that have been used for brewing beer, on the other hand, the cells clump together and fall to the bottom. This is how your beer turns clear. Although these days they use like centrifuges to speed up the process. But the reason why your beer is not really cloudy is that the yeast strains used to brew beer form what we call flocks. They are flocculent, meaning the cells clump together into little lumps, and the little lumps fall to the bottom, fall out of suspension. So some yeast strains, if you grow them in liquid, they'll flocculate and fall to the bottom. Other strains will stay separate. The individual cells will not clump and they don't fall to the bottom. And it turns out that flocculation is governed by a single gene called FLO1. Yeast cells that have it have a cell surface protein that binds to the cell walls of other cells. Okay, so we have a gene that causes yeast cells to flocculate. How is this a green beard gene? Well, it turns out that flocculent yeast can survive chemical shocks much better than non-flocculent yeast because the flocculent yeast shield each other. If you zap flocculent and non-flocculent yeast with the antibiotic amphotericin B, almost none of the non-flocculent yeast survive. Almost all of the flocculent yeast survive. Same is true for hitting them with hydrogen peroxide. None of the non-flocculent yeast survive. About 80% of the flocculent yeast survive. It's not quite as obvious, but you can see a similar effect from exposure to 10% ethanol. That will kill about 50% of your non-flocculent yeast, but only about 10% of your flocculent yeast. And flocculent yeast tend to stick to other flocculent yeast, all each other, and they form these flocks where the yeast on the outside get killed, but they shield yeast on the inside. So that's a flock right there that has been sliced in two, and these flocks have been stained with a blue stain that stains dead cells. So there's a regular flock. Two right there, there's that antifungal drug, and you can see there's a thin blue rim. There's a rim of dead cells, but that rim is incorporating a very big part of the flock that's made up of live cells, the part here that isn't stained blue. So the cells on the outside of the flock died, but by dying, they've protected the cells on the inside of the flock from dying because they block that antifungal drug. You get the same effect with exposure to 70% ethanol for 5 minutes. You can see there's that blue rim. Those are the cells that died so that the cells on the inside might live. With longer exposure to ethanol, you can see that more of the cells die as the ethanol diffuses in, but the cells on the outside have still protected the cells on the inside from being poisoned by ethanol. So this is a green beard gene. If you're a yeast cell and you have the flock gene, first of all, that makes you, that gives you a cell protein, that's the trait, that makes you flocculate with other flock yeasts. So yeasts that make this protein flock together. And when yeasts that make this protein are flocking together, they can survive chemical attack. A few on the outside will die, but most of these cells will survive. That's a green beard gene. It controls a trait, which is the presence of that cell surface protein, and it causes all yeast cells that have that trait to behave altruistically towards each other, flocking together in these cottage cheese-like flocks, these cottage cheese-like lumps. So that's a real-world case of a green beard gene. Okay, I tell you what, I've got 11 o'clock. That's actually a pretty good place to take a break. I'll leave you to contemplate that, and we will take about 15 minutes and reconvene at 11:15. And I will stop recording